Bismillahirrahmanirrahim. Assalamu alaikum. In this video, we are going to discuss peptic ulcer pharmacology. So what is peptic ulcer disease? Well, peptic ulcer disease is actually the erosion of the mucosal lining of those parts of the GIT that are exposed to the acid pepsin mixture. So this acid pepsin mixture, it causes ulceration in the mucosa of the stomach, or it could be the first part of the duodenum, or it could be the lower part of esophagus. Individually, they are named as stomach ulcer or duodenal ulcer. And when we talk about peptic ulcer, we are actually talking about all of them. Now, to understand the rationale of the treatment of peptic ulcer, we have to understand the factors that are responsible for causing peptic ulcer. So we have to understand two very important factors. One is the aggressive factors and the other one is the mucosal defensive factor. So you can see that the mucosal integrity is dependent upon the balance between these factors. By protective factors, we mean that all those factors that are preventing the erosion of the mucosa, while aggressive factors are all those damaging factors that are responsible for causing ulceration. So you can see in this figure, we have the gastric lumen where we have the acid pepsin mixture, which is used to digest food. This is an aggressive factor because it can also digest the gastric mucosa, but it cannot do so. Why? Because of the mucosal defense, in which mucus production is one of the components. So in a healthy subject, there is a balance between both of these factors, and as a result, this acid pepsin mixture does not digest the mucosa and does not cause peptic ulcer. So whenever the aggressive factor dominates, it results in peptic ulcer. So now in this figure, if we look towards the aggressive and damaging factors, first of all, we have hydrochloric acid, and then after that, we can see pepsin, and then we have a very important factor, which is NSAID, non-steroidal anti-inflammatory drug. Those patients that use this continuously. Along with it, smoking also is a factor which is responsible for damaging the mucosa. And one of the very common factor responsible for peptic ulcer is H. pylori infection. And then we talk about the defensive factors. We can see in this figure, those have been mentioned, starting with bicarbonate. Bicarbonate is a very important defensive factor. As we know that the pH of the stomach lumen or acid is in between 1.5 to 3.5. So whenever this acidity starts increasing more and more and the mucosa becomes endangered, in that case, a lot of bicarbonate is released to neutralize this acidity. Another important factor, defensive factor, is the mucus, which is secreted by the gastric epithelium, and you can see in this figure that it prevents the contact of the acid pepsin mixture with the epithelial cells. So in between the epithelial cells and the HCl in the gastric lumen, we have layer of mucus and then bicarbonate secretions. You can see that the bicarbonate is actually staying in between the mucus and the epithelium, and hence it neutralizes the excessive acidity. You can see at this point where we have bicarbonate, the pH is high, which is around 7, while the pH in the gastric lumen is very low, usually between 2 or 3. And then another important factor is good blood flow, which is very necessary because if ischemia occurs, that can also lead to mucosal ulceration. So the main aim of the pharmacology will be to maintain the balance to treat the ulcer. So now we are going to discuss the gastric glands and the types of cells present. First of all, we are going to talk about the parietal cells. You can see in this figure. And parietal cells, they are actually the main thing that we are going to discuss in this lecture, as it is responsible for secreting hydrochloric acid. Then the second type of cell is the chief cells that are responsible for secreting pepsinogen. So you have to also remember that pepsin is the active form and pepsinogen is the inactive form. Pepsinogen gets activated into pepsin at a low pH, that is near to 2, around 2, and at high pH it becomes inactive. And then we have the G cells. G cells, also known as gastrin cells, they are also present in the duodenum and pancreas, and they are responsible for releasing the gastrin hormone, which stimulates acid production. And then you can see we have enterochromaffin-like cells. These cells are also very important because they release or secrete histamine, which is responsible in stimulating the parietal cells to secrete HCl. So now you can see in this figure, we are going to discuss how parietal cell is going to secrete HCl into the gastric lumen. Now you can see in this figure the parietal cells, it has very important receptors. The first one you can see is M1, which is the muscarinic receptor. The second one is the G receptor where gastrin acts, and the third one is H2 receptor, and we have also fourth one which we don't have in this diagram, but that is prostaglandin receptor. So all of these together are responsible for HCl secretion. So if any one of those receptors is stimulated, it is responsible for enhancing the HCl secretion by the parietal cell. So the three receptors that you have in this figure are responsible for stimulation of HCl, and prostaglandin receptor is opposite to the three of these. Now let's discuss them one by one. First of all, talking about M1 receptor, which is muscarinic receptor. It is stimulated by acetylcholine as a result of parasympathetic stimulation, which stimulates the enteric plexus and ultimately the vagus nerve, which binds with M1 receptor and is responsible for HCl secretion. So now it's easy for you to understand that parietal cells, they are also supplied by the vagus nerve, and hence vagal stimulation will result in HCl secretion. So as a result of the vagal stimulation, acetylcholine is secreted. That acetylcholine, not only it binds with the M1 receptor, but it also stimulates the G cells, and the G cells ultimately secrete gastrin that bind with the G receptors present on the surface of parietal cells. Now as a result of this vagal stimulation, which results in secretion of acetylcholine, and then acetylcholine stimulating the G cells to release gastrin, this gastrin and acetylcholine also stimulates the ECL, the enterochromaffin-like cells, which are very important because they are responsible for secretion of histamine, which act on H2 receptor present on the parietal cell. So as a result, the stimulation of H2 receptor is the final pathway. So as a result of the stimulation of H2 receptors, it stimulates the cyclic AMP. You have to keep in mind, when cyclic AMP is stimulated, it results in stimulation of a lot of proteins in the cell known as protein kinases. So in the parietal cells, a series of chemical reactions takes place. water and carbon dioxide combine in the presence of carbonic anhydrase enzyme to form carbonic acid. This carbonic acid further splits into hydrogen ion and bicarbonate. The molecule of bicarbonate, it enters into the blood, while the molecule of bicarbonate, it enters into the blood, while the proton will leave or secrete from the parietal cells with the help of a pump known as proton pump. This proton is secreted into the lumen to be a part of the gastric acids in the lumen. So this proton pump is known as hydrogen potassium ATPase pump, which allows the proton to be secreted in the lumen of the stomach, as you can see in the figure. This proton is secreted into the lumen of stomach in exchange of potassium ion. So the name proton pump is due to the fact that its function is to pump proton into the lumen of the stomach. Now the important thing to remember is that this proton that is secreted into the lumen of the stomach combines with the chloride in the lumen, and it's not secreted together from the parietal cells. Now from where this chloride ion comes, you can see that this chloride ion also comes from the parietal cells via several mechanisms. So this chloride ion enters the parietal cell in exchange of the bicarbonate ion. So this means that the proton and the chloride, both of them, come from the parietal cell from different mechanisms, and ultimately they combine in the lumen of the stomach to form HCl. So now talking about the fourth receptor, which was very different in its function. Prostaglandin is responsible to decrease the HCl secretion. You can see in this figure that the stimulation of prostaglandin E2 receptor inhibits the cyclic AMP production, and ultimately it inhibits all the further reactions that take place as a result of activation of cyclic AMP that we have discussed right now. Not only decreasing the HCl secretion, but prostaglandin also increases the protective mucus layer production. As you can see in this figure that it enhances the mucus production. So it acts as a cytoprotective agent. And the third important function of prostaglandin is that it causes vasodilation. By causing vasodilation, it enhances the blood circulation, and if there is any kind of erosion, it will help in its immediate healing due to the increase in blood flow in the gastric bed. So these three important functions of prostaglandin you should remember.